All right, everybody. So we'll get started. I've been giving a more abbreviated intro recently, but we have some great attendance here, which is fantastic. So I'm going to give the sort of the full-on intro. So hi, everyone. My name is Robert Driver. I'm a postdoc at Duke University, and I want to welcome you to the AOS Student Research Grant Seminar Series. And so before we get to our speaker today, I want to give an introduction about the seminar series, and also an introduction about the AOS Student Research Grant, which the seminar series stems from. So each year, the AOS offers funding to students and early career researchers with the AOS Student Research Grants. And each year, the AOS receives over 100 applications. There were almost 200 applications this year, actually. So making these highly competitive, even compared to other society-based small research grants. So I really want to acknowledge the substantial amount of effort that goes into writing a successful proposal. Winners have to describe a project that advances the field of ornithology while still remaining feasible within the AOS Student Research Grant budget, often as part of a graduate dissertation. So the AOS Student Research Grant seminar series celebrates the challenging and innovative work of the AOS Student Research Grant recipients by sharing AOS funded or related work to the AOS members. So we give out this money or the AOS gives out this money and funds some fantastic projects which we often don't hear about again. So the AOS Student Research Grant Seminar Series lets us really appreciate all the hard work of the AOS members. So each week we'll showcase the research and research plans of a 2023 or 2022 AOS Student Research Grant winner in a seminar style format. And we'll have one seminar each week for 15 weeks until May 5th, and we are right at the halfway point. This is our eighth week. And I want to thank everyone for being here. I want to thank the AOS and the AOS Student Affairs Committee for support and input, and especially thank Chris, our speaker today. So before we turn our attention to Chris's research, I just want to mention to please put your microphones on mute. If you have questions, there will be time at the end for questions. During our Q&A time at the end of Chris's talk. And you can participate by raising your hand and I can unmute you, or you can use the Q&A function in the Zoom window, or I'm going to enable the chat for all participants in a moment, and you can also post your question in the chat. So many different ways that you can post your question. We'll check all of them and make sure we cover your questions. So I'd like to remind everyone that this is an AOS sponsored event and that all participants are required to follow the AOS virtual meeting event code of professional conduct. And I will post a link to the AOS virtual meeting event code of professional conduct in the chat. And please be aware that we are also recording this talk. So I'm also going to post the contact information for the Student Affairs Committee if you have any questions about the seminar, as well as a link to the post-seminar evaluation form. I just want to emphasize this because we have a larger audience this week. If you can take the time to fill out the post-seminar evaluation form, that really helps us in this series of seminars as we're trying to improve it as we go along here. So I will post those links in the chat as soon as I'm done speaking here. Okay, so today, Chris Sayers is currently a PhD candidate at UCLA. And Chris's academic career began with a BS in Environmental and Sustainability Sciences from Cornell University. And this is where Chris got his first experience researching the impact of mercury on birds, studying the geographic patterns of mercury in breeding tidal marsh sparrows. So I'm guessing that's Nelson's or Seaside or something like that. Chris's BS research is published in Ecotoxicology. And after graduating, Chris continued to work with birds and mercury exposure as a research biologist at the Biodiversity Research Institute in Portland, Maine. In 2022, Chris joined Morgan Tingley's lab at UCLA, where he continues to research mercury exposure in birds, this time as a result of pollution from gold mining in the neotropics. And in 2023, in addition to receiving the AOS Student Research Grant, I just want to mention Chris also received the Sigma Xi Grant in AIDS of Research Award, which is a challenging award to receive. And 2024 was a big year for Chris. Chris was awarded two major and prestigious awards, the Future Investigators in NASA Earth and Space Science and Technology Award. And secondly, after receiving honorable mention in 2022, in 2024, Chris received the NSF GRFB, the Graduate Research Fellowship from NSF. And with that, I will turn it over to Chris for his talk today. The Canary in the Gold Mine, assessing impacts of artisanal and small-scale gold mining on bird communities in the Peruvian Amazon.
Well, thanks so much for that introduction, Robert. And thank you to the AOS for not only hosting this awesome seminar series, but supporting the work that I'll be sharing today. I'm going to be presenting some preliminary results of our field campaign to assess the biodiversity impacts of artisanal and small-scale gold mining in the Peruvian Amazon. And we are leveraging birds as effective indicators of ecosystem function and pollution exposure to be able to do that. And I want to give a quick shout out to my advisor, Morgan Tingley, who is celebrating his birthday today. So happy birthday, Morgan. All right, so a brief outline for today. This talk,
Chris, sorry,
yep,
you just got to share your screen is unshared now, at
oh, it's
least for
unsure.
me.
Okay. Is
Does
it
anybody else have
back?
that issue?
Is it back?
I've got it now. I'm sorry
Okay.
if that was just me. I apologize.
Okay, no worries. Okay, so yeah, this talk has three distinct sections. We'll begin with a robust background of the study system, followed by the first two chapters of my PhD dissertation. I'll be sharing a lot of information over the next 45 minutes, roughly. So if you get lost, you have this ticker tape at the top to orient yourself, okay? And so we'll get started with some background. So I'd like to begin by introducing the setting for this talk, Amazonia. The Amazon is the world's largest tropical forest. It recycles around 15% of Earth's fresh water and also supports over 10% of all species on Earth. And so it's a pretty special place. It's therefore one of the most crucial biogeographical regions to achieve international conservation and sustainable development goals. But rapid land use change is putting the system in jeopardy. This is a very Brazil-centric time lapse, but the red area here illustrates forest loss from 1997 to 2015. And since the 1970s, the Amazon has lost roughly 1/8 or 13% of its original forest cover due to agriculture expansion, mostly for beef production and resource extraction as well. But it's not just the raw land use change here that's concerning. It's estimated that an additional 38%, or roughly 25 million square kilometers of the remaining forest has been degraded by associated processes of selective logging, fire, edge effects, and drought. And it's really the sum of this habitat loss and degradation that's threatening the region's biodiversity. And one of the primary examples here that we can turn to is birds. And in large part due to the biological dynamics of Forest Fragments Project, or BDFFP, just north of Manaus, Brazil, we now understand how poorly tropical forest bird communities are responding to forest degradation and fragmentation. Species that eat insects and spiders and utilize forest understories, collectively understory invertivores, as I'll be repeating a lot of times throughout this talk, typically decline faster than other groups in forest fragments due to really a variety of life history traits. They have restricted thermal niches, which makes them less tolerant to temperature changes as after you remove trees, but also under global climate shifts. But their poor dispersal ability is thought to be one of the most limiting factors. It's really difficult for them to cross forest gaps with their rounded wings and weak pectoral muscles. So we can see pretty instantaneous drops in survival post-isolation of forest fragments. Surprisingly, though, several long-term bird monitoring programs across Latin America, not just in Amazonia, over the last decade have noted steep understory invertivore losses in undisturbed lowland sites in Ecuador, Costa Rica, Brazil, and Panama. And this has sparked sort of broad speculation of potential drivers here. Many researchers have pointed to climate change as it produces both thermal stress and bottom-up stress via declines in insect prey stocks. And thanks to Jared Wolf and colleagues, we finally have empirical evidence for this. So they analyzed over 27 years of bird captures at the BDFFP study plots, again, north of Manaus, Brazil. And they predicted that with a 1 degree Celsius increase in average dry season temperature, that would reduce the mean apparent survival of the understory bird community by 63%. So that's quite significant. And a similar trend existed for reducing precipitation as well. So climate change here is clearly a driving force behind bird declines in undisturbed forests. But as with many conservation dilemmas, we don't expect climate change to operate in isolation. And there are other factors that are changing across the Amazon basin that may add additional stress to birds. And so throughout this same time series, the price of gold has skyrocketed to an all-time high. It started this ascent following the economic crisis in 2008. and it's currently trading at $96 per gram as of yesterday. And literally every time I check this number, the stock is up a few dollars. We are seeing some truly unprecedented growth here, especially within the time of my PhD. And this tremendous economic opportunity has produced an ongoing gold rush throughout the Amazon and other tropical regions, in which artisanal and small-scale gold mining, or ASGM, operations are targeting alluvial gold deposits in riverbeds and adjacent soils that have eroded down the Andes Mountains over thousands of generations. But the factor that I think makes ASGM stand out from other drivers of land use change in Amazonia is that it's also the largest anthropogenic source of environmental mercury pollution. So this accounts for around 40% of global emissions to air. And this proportion is way higher for Latin America, pushing 80% approximately. And the reason this is because miners are regularly using mercury as an inexpensive way to extract gold particles from sediments, creating these mercury gold amalgams on the right there. And the growth of ASGM is why we continue to see increases in global mercury emissions despite relative pollution reductions, relative pollution wins in the global north. Sorry, my room keeps dimming, so I'm going to do that quite frequently, I think. Why is this a problem? Mercury is a persistent environmental pollutant that adversely impacts human and wildlife health on a global scale. It is a naturally occurring metal. It is released often by geogenic sources like volcanoes and hydrothermal vents. But anthropogenic activities have greatly enhanced global concentrations in soil, water, and air. Once present in the environment, microbes can convert inorganic mercury to methylmercury, which is a much more toxic and bioavailable form that readily bioaccumulates in individuals and biomagnifies up aquatic and terrestrial food webs. And so methylmercury poses direct health threats, reduced cognitive function, immune response, reproductive success, as well as behavioral and developmental defects, especially for long-lived individuals foraging at higher trophic levels. And so throughout this gradient, our mercury risk here is increasing exponentially. And the reason I find mercury so dangerous is because it's inconspicuous in nature. It's not something that you can interact with in trace quantities, right? You can't see it, you can't taste it, you can't smell it, which makes it really easy to overlook. But the reason it's particularly relevant here in Amazonia is that the basin accounts for almost a third of the total inorganic mercury global land sink. And similar to carbon sequestration, trees are really excellent at scavenging gaseous elemental mercury out of the atmosphere. And so we can see a signal for this large mercury flux within bird communities of the region. From the synthesis that I led on bird mercury exposure across the neotropics back in 2023, we know that ASGM elevates mercury bioaccumulation across avian trophic niches. So bird mercury concentrations were over 4 times higher at sites impacted by ASGM. We're looking at the difference between circles and triangles in these means here. And in this study, we detected some of the highest concentrations that have ever been documented in bird taxa. And this translates to elevated conservation concern for this same functional group, the in steep decline, the understory invertebrates, right? Especially for families including wood creepers, wrens, and antbirds. And so this apparent overlap between elevated mercury bioaccumulation and the losses of understory invertebrates brings us to a very simple question. How does mercury, specifically from gold mining operations, contribute to biodiversity loss? And maybe along the same lines, how may mercury exposure contribute to ongoing declines of the understory invertebrates here in Amazonia? But while this question is pretty simple in form, the reason it's difficult to answer is because ASGM integrates many other drivers of biodiversity loss, all happening at the same time and place. And so I'm going to walk you through, to the best of my abilities, how this process works. As I said, ASGM operations are largely targeting alluvial gold deposits in soils and sediments within floodplains. So miners that arrive by boat may start at the river edge, but typically expand into adjacent habitats by removing trees, especially if they have a good product yield, leading to deforestation. More gold attracts more miners and equipment to process more sediment. And so typically miners use hydraulic pumps to either suck up sediment or blast away new sediment, which leads to drastic hydrological change as miners divert rivers and develop their networks of mining ponds. After isolating some of the fine sediments, miners regularly use mercury, almost like a magnet, to amalgamate those suspended gold particles in their mining mixes. And informal and illegal operations, which account for the majority of the industry, often discard the excess material directly into the environment. Now, once in the environment, local microbial communities, again, can regularly transform this inorganic mercury into methyl mercury, which then works its way into local food stocks. And largely, indigenous communities that rely on fish stocks are bearing the largest health costs of this. Now, miners then take their gold mercury amalgam and burn it over an open flame to isolate the gold nugget. And so, of the total mercury in your average ASGM operation, it's estimated that 40% gets discarded into water and soil. Where does the other 60% go? Directly into the airspace. You also have this sort of feedback to the miners themselves in terms of direct skin exposure, but also mercury inhalation during this process. So not a good thing. And from there, it's really up to the local weather patterns to determine its fate. But we know that canopy trees adjacent to artisanal gold mining operations scavenge a great portion of this deposition and out of the atmosphere. And this mercury comes down to forest floors with precipitation and leaf litter fall, where it can be methylated, bioaccumulate, and biomagnify within terrestrial food webs. But that's not all. These direct environmental disturbances of ASGM can lead to several other indirect consequences, right? So forests adjacent to mining zones also face greater access and exploitation by humans, which facilitates greater wildlife overexploitation and poaching in some cases through bushmeat hunting and trapping, et cetera. And in creating more stagnant water, ASGM drastically increases the amount of breeding habitat for disease vectors like mosquitoes, which leads to increased avian malaria and also malaria to humans in mining landscapes. So distilling everything down to this concept map, right? We expect that all of these disturbances will have a negative impact on biodiversity in their own right, but also have the capacity to amplify one another through a variety of feedbacks. So the solid arrows here represent relationships grounded in the literature, and the dotted arrows indicate predicted relationships based on our current understanding. And so if we want to understand how mercury pollution as a unit may be impacting biodiversity, we need to first separate its impacts out from a plethora of other environmental disturbances. Now, this has all sort of been very cartoonish to sort of try to distill a lot of the complexity here, but I want to give you a sense for what these systems actually look like on the ground. So this is a photo of a typical artisanal gold mining production. We have a barge carrying a suction pump floating on a mining pond, which is pumping water up to their sluice, that elevated chute at the top of the frame there. Mechanically, it's really not that complicated of a process. You can see how few items that are actually necessary to extract gold. But mining is very cyclical, right? So after expending the sediment in one area, miners pack up their things and move over to the next, which is often just a few meters away. So it creates this matrix of sand and mining ponds, essentially an artificial desert. And without the trees to shade the landscape, the sun bakes this sand matrix to around 40 degrees or even higher degrees Celsius, that is. Without the soil nutrients, it's really difficult to kickstart the typical forest regeneration process. And some of the pioneer plant species actually really struggle in these landscapes. So though the industry is called artisanal and small scale gold mining, the cumulative impact of these tiny mining cycles is actually quite large. It's estimated that ASGM contributes roughly 10% of annual forest loss in the Amazon, though I have to admit that the data on that are not fantastic. But undoubtedly it has disproportionate impacts in specific regions. So one of those being Madre de Dios, Peru, in the Western Amazon. The photo on the left is an aerial image of one section of Huepetue, one of the largest gold mines by area in the world, at least in South America. And it also produced 2% of the total gold in 1998 in its peak production. The GIF on the right is La Pampa, one of the largest illegal gold mining operations in the Peruvian Amazon, which effectively tripled in size between 2011 and 2023. There's a ton happening in these two photos, so I just want to take a minute to explain. The rapid growth of these mines is attributed not only to increasing gold price, but also to the completion of the Interoceanic Highway in 2011, which runs from west to east in that right GIF there. So we can see this expansion of ASGM, which are the pale scars in the center. But also note that the rivers change route along the south, or like the bottom panel of this frame, and that's the Rio Malinowski. And it gets definitely more direct via the amount of water that's being changed by the miners themselves. I also want to point out that there's a ton of small forest clearings that are popping up along the roads that run perpendicular to the highway. Some of them connect to the mines themselves. So it's an intense amount of landscape change. And so since 1984, ASGM activities in this region of Madre de Dios have consumed nearly 100,000 hectares of forest, which is an area 25% larger than New York City. Madre de Dios, Peru was already a well-recognized hotspot for biodiversity, but the recent growth in ASGM has catapulted it as a hotspot for ASGM as well. And so I've been fortunate enough to have the opportunity to collaborate with the Center for Amazonian Scientific Innovation, or CINSIA, an NGO based in Puerto Maldonado, the capital city of Madre de Dios, to conduct this research. And to give you some brief political context, Peru is the largest exporter of gold in South America. It has 43% of this export is illegal gold, which now surpasses the revenue of cocaine. And in 2016, the 46,000 miners that work in Madre de Dios produce 11.5% of Peru's total gold export. So it's an intense amount of work and export that's going on there. At the same time, though, a third of Madre de Dios is protected as either conservation or indigenous lands, and it supports among the highest alpha diversity on the planet. So it serves as a really great platform for the body of work that I'm proposing. So that brings me to chapter 1, where I'm asking the question, how does avian diversity and mercury exposure vary across protected and mining sites? And so I've chosen to think about this system in terms of disturbance gradients, plural. The first disturbance gradient that we can instantly recognize here is that ASGM operations usually create this large edge effect, where the influence of deforestation really decreases asymptotically as you move deeper into forests. And if we could leverage this effect to compare avian diversity and mercury bioaccumulation at set distances from forest edges, we would then have we would be able to simultaneously account for the influence of these stressors at a given sampling location, which would then give us the opportunity to isolate their respective impacts on birds. But to actually observe an effect requires another disturbance gradient and sampling sites with different proportions of mining-induced disturbances. So looking at the broader picture of the gold mining industry, it essentially operates on two main axes, at least from my perspective. The first runs from formal operations to illegal operations. Upwards of 70 to 80% of global ASGM operations are either informal, for example, lacking permits or land titles, or illegal, for example, mining protected areas with forced labor. The other axis runs from large scale to artisanal mining. And the main elements that are changing along this axis are money or financial investment and the amount of soil processed per day. And so to highlight two main case studies, we have Yanacocha, Peru, and in northern Peru in the top right quadrant, which is the fourth largest gold mining operation in the world in terms of gold production, and it's responsible for the majority of Peru's export. And we have La Pampa on the bottom left quadrant, which is itself an illegal operation, but has also been associated with all sorts of other legal activities like narcotics and human trafficking. And so in this study, to reduce the security risks in the field, we'll be focusing on mining operations that fall primarily in the top left quadrant, approximately here. And so we are sampling 3 land use classes in this case, including conservation reserves, small scale gold mining, and artisanal gold mining operations to attempt to isolate the effects of mercury. Small scale gold mining is similar to artisanal mining in that they both target alluvial gold deposits, but small scale mining is much more profitable and has a different land footprint since they can afford to process more soil per day. They involve typically more sophisticated equipment, like heavy machinery, and can afford to process the gold a bit differently than artisanal mining. So given these differences, I'm hypothesizing that small scale gold mining will have less hydrological change, less mercury emission, and less hunting than artisanal operations, largely because in having more capital, they can afford to export their mercury amalgams, say, to gold shops in town away from the mine itself, and also import food items to the mining sites themselves. And so just as a sidebar, I'll be continuing to use these colors, blue, yellow, red, to represent these land use classes. So this is a slide to just help us visualize those sort of finer differences between small scale and artisanal mining. You can see that the land disturbance on the left is fundamentally different. Miners are using heavy machinery like bulldozers and excavators to actually remove and excavate the land rather than just sifting the surface with suction pumps. And so this all sort of brings us to a visual schematic of our sampling design. And I want to emphasize here that this is the sampling design. This is the system that we've set up that's going to supply data for all of the work that I'll be talking about today and for the remainder of my PhD. We have conservation reserves on the top again, followed by small scale gold mining in the center, followed by artisanal mining on the bottom. And this is an effort that integrates several different sampling techniques. We use bird banding and passive acoustic monitoring to measure how avian diversity and mercury exposure are changing along standardized transects that feature a continuous array of mist nets from open mining zones through forest edges and into forest interiors. What's not pictured here is that we're also deploying passive air samplers that along this gradient too to measure inorganic mercury emissions in the airspace, to sort of account for what the deposition might be in the landscape. And via this design, we have the opportunity to monitor bird responses to these respective stressors at a reasonably fine scale across these two disturbance gradients. So to now zoom in on what's happening on the ground, we use mist nets to capture individuals from the understory bird community. We log a variety of morphological and demographic data for each individual. But most importantly, we're extracting blood and feather samples for later mercury analysis. I also deploy Swift 1 autonomous recording units to continuously record entire soundscapes 24 hours a day, more or less for 2 weeks straight. And so this produces a ton of information on animal, but also anthropogenic sounds that we can leverage for later questions. I won't have time to get into this today, but I'm happy to discuss these data further in the Q&A. So this field sampling involves the help of over 20 collaborators and assistants, most of which are undergraduate biologists. And it has been a real honor to get to serve as one of their mentors, just to be a part of this entire process. So far, my role on the team is usually as lead blood sampler. The students usually call me El Vampiro, or the vampire. So I guess that's the main field skill that I'm bringing to the table. Peru works a little bit differently than the US in that to be really taken seriously as a biologist, you need to publish a thesis to acquire your biologist title. So I'm in the process of working with a few of these students to help them reach this next step, which is really exciting. And to give you an idea of how much we've been able to accomplish today as a team, after about 9.5 months of fieldwork since 2023, our team has surveyed 14 sites. We've operated over 1,200 mist nets, amassing over 13,000 net hours. And this effort has produced almost 2,000 bird captures across 200 species. And our ARUs are collecting or have collected over 20,000 hours of soundscape recording. So I think we're at something crazy like 12 terabytes. And that number is supposed to go up. So it's an incredible amount of processing power that we're going to need. So on the right is a rough map of our sampling sites. Notably, this does not include everything to date. We've added a few control areas to the east of that Inca Terra Point and also to the north in sampling Cochacashu just this last February. And really the primary objective heading into this upcoming field season in 2025 is to sample more mining sites in La Pampa. And so using this roster of capture data, I've been starting to pull out some of our preliminary relationships, and I'll be presenting all of the community data to date, but only mercury data from 2023 and part of 2024. We're still waiting on some of the 2024 samples to come back from the lab. But since we're mostly a bunch of bird nerds here, I figured I would talk to y'all about some of our most interesting sightings. So this is a Gueldes antbird. On the left is what a typical male looks like. And on the right is a bird that we captured at Causai Biological Station on the lower Madre de Dios River, east of Puerto Maldonado. It's displaying signs of leucism. And from what I can tell, I haven't been able to find any evidence of this in other individuals. So it's a pretty neat finding. Up in the ante in terms of rarity, we caught a black bushbird in a small-scale gold mine in Huepetue, and it's a reasonably rare species in the region. There's fewer than 800 total sightings in eBird. So I nearly fell off my chair when this one popped out of the bag. This was my lifer. So definitely some of the perks of field work. Up in the ante in terms of rarity again, this is an olive-spotted hummingbird, and its typical distribution is along the sort of larger tributaries of the Amazon basin in Ecuador and Peru. They forage along riparian floodplains in dense heliconia patches. And so we caught this one in an artisanal mine in Labyrinto, which is roughly here where that red arrow is pointing. This is not the first record for Madre de Dios. It's sort of a gradual evidence of range expansion. And here's a picture of that tail pattern in case any of you thought that I was misidentifying this for a female fork-tailed wood nymph. Another hummingbird that's well outside of its range is we've actually caught 3 buff-tailed sicklebills to date. One was the lowest elevation record to date in La Pampa. Yeah, a really degraded habitat. And I think part of the point of why I'm showing you these individual sightings is that going into these places for the first time in 2023, I didn't really know what to expect. I mean, these are highly degraded systems, but to my personal surprise, they actually hold a great deal of life. So, and that red arrow is pointing to where we caught that bird. Saving the best for last, by far the rarest bird we've detected in this study happened outside of a gold mine in Cochacashu Biological Station just this past February, which is roughly here. This is a globally rare species, the Andean lanisoma. It's the lowest elevation record that I've been able to find in eBird, and also only the 48th sighting to date. And so this species is thought to sort of occur in these low-lying ridges of the Amazon, sort of in montane systems, but this was flat out in the lowlands. So it's long been thought that this species displays altitudinal migration by season. So I don't know if its sighting actually supports that or not. I guess we'll need more sightings to actually confirm that, but definitely a huge surprise. And, you know, Madre de Dios has been one of the most well-studied tropical places on Earth, but clearly there's still some things left to learn about the system. So now jumping into the quantitative results here. These figures are hot off the press. Not even my lab mates will have seen the majority of the data that I'll be presenting today. And we'll start by talking about mercury, which I plot on the y-axis, the vertical axis, and as a function of bird trophic niche on the x or horizontal axis. And so far, there haven't been too many surprises here. These results are a nice complement to our 2023 synthesis. We see that birds that eat plant matter on the left have significantly lower mercury concentrations than those that exclusively eat insects and fish on the right. And so I'll point out that many of these data points are above the red dotted line here, which indicates the EC10, or the concentration that equates to a 10% reduction in reproductive success at 2.4 ppm when analyzing flank feathers. We'll have the opportunity to talk about some of the nuances there in a second. But I think another key takeaway from this figure is that we're collecting a high proportion of our samples via our undersort invertivores. And so that's really helpful since they are sort of the conservation priority here. And so I could cut to the chase and start talking about these understory invertivores, but I know that some of you want to talk about some of the craziest numbers in the system. So we'll be hosting our medal ceremony for the highest mean concentration at the Mercury Olympics. Our bronze medal goes to the green rufous kingfisher with a mean mercury concentration of 22.8 ppm. The silver medal goes to the green kingfisher, a smaller species with 26.2 ppm. And our gold medal goes to the behemoth Amazon kingfisher with 36.9 ppm. The single highest concentration of this entire study, though, setting a new record for the highest mercury concentration in South America, goes to the smallest piscivore in the Americas, the American pygmy kingfisher, with a concentration of 80.6 ppm. So we captured this guy, this male, at an artisanal gold mining operation just outside of Labyrinto, Peru. But it's an insane amount of pollution that these birds are harboring. And so for the remainder of the talk, we'll be focusing on the understory invertivores. And so our bronze medal goes to the white-throated antbird. Silver medal goes to the musician wren. And the scaly-breasted wren gets our gold medal with 9.6 ppm. And so to get a sense for how risky these concentrations are for these species, unfortunately, we don't have a mercury effect threshold that has been published for a neotropical bird species yet. And so we have to extrapolate these concentrations from the global north. There's some limitations for that, but it's sort of the best we can do at the current time. And so in this case, I'm using pretty well cited thresholds from the Carolina wren by Jackson et al. In 2011, where yellow represents a 10% reduction in reproductive success, orange represents a 20% reduction, red represents a 30% reduction, and black represents a 40% reduction in reproductive success. And so we're finding that some of the most at-risk species here are either obligate or opportunistic army ant followers. And this is really a trend that has been on my radar for quite some time, especially in our data sets. And my standing hypothesis right now is that these species may have disproportionate access to high mercury prey items like spiders, but I really welcome any potential hypotheses from the audience. Next, we want to focus on where on the landscape these birds are being exposed to these high levels of pollution. And so breaking down mercury concentrations by land use class, we find that birds utilizing forests next to mining areas have the highest exposure, which is not a surprise. It's definitely in line with our hypothesis, but we currently are not detecting a significant difference between small scale and artisanal gold mining. This effect actually was significant using our 2023 data, but it has since dropped off after incorporating the 2024 data set in a linear mixed effects model framework. And we'll see what happens after this summer, obviously. But I do want to highlight, though, that this, in terms of a risk assessment, the mean and median of the concentrations coming from artisanal gold mines are above that EC10. So moving to an even finer spatial scale now in leveraging our mist netting transects, we're detecting significantly higher mercury concentrations in forest interiors, but specifically next to artisanal mines. And now this isn't, the effect size here isn't huge. And obviously we'll see how things change as more data continue to trickle in from the lab. But to me, this result is potentially our most concerning, since it suggests that these sensitive understory invertivores are experiencing the most toxic risk in areas of the forest that should be refuges, canonically speaking. But so the big question that sort of remains is, what impact is all this mercury having on populations and communities? And so to get an idea of how bird communities across sites and habitat types compare to one another, I calculated the Bray dissimilarity across sites and ordinated them in 2 dimensions using non-metric multidimensional scaling. In this plot, the axes are essentially meaningless, but each point represents a site, and the relative distance between each point represents the relative similarity of their bird community. And so different colors here represent different land use classes, and different shapes represent different habitat types. And so far, we're not finding any significant difference between our land use classes, the difference between blue, yellow, and red points, but a strong significant difference between communities occupying primary forest and successional habitats. Circles on the left and triangles on the right. And so this plot suggests that land use change, not mercury, is the dominant driver of community turnover across the landscape, which is definitely not super surprising, right? That's sort of in line with our expectations. And so to better visualize this shift from left to right, primary forests with understory species like this band-tailed manakin, a fruit-eating species, are being converted to open areas with lots of water, which can be exploited by birds like this green kingfisher, a fish-eating species. Zooming in again on the primary, or at least formerly primary forest, things get a bit more interesting. And so when we plot the accumulation of unique species as a function of distance from forest edge, we find that there are 40% fewer invertivore species along transects in artisanal mines as compared to reserves and small scale mines, the blue and yellow versus the red lines. And so the reason I doubt that this is a purely an effect of deforestation is because there are superficially similar rates of selective logging in both mining classes. And so it certainly begs the question, is mercury potentially playing a role here? And so in this last plot, we are finding that there's actually a strong negative trend between species richness, which is really just one of the many indices of biological diversity that we can use here, and mean concentrations, mean mercury concentrations at the site level. And so I was originally quite hesitant to show this trend after my first field season, since I thought it may have just been an artifact of poor sampling, but I am seeing this trend get actually stronger over time as we incorporate more sites. And this is an incredibly concerning finding, right, from a conservation perspective. But we do need to bear in mind here that it's sort of a classic case of correlation versus causation. This plot does not yet consider the habitat structure at each of these sites, which could be collinear with mercury pollution. And so that's one of the things I'll be focusing on in the later parts of my PhD to be able to disentangle that. So in general, these results support many of my principal hypotheses and give us a lot more confidence that this sampling design is in fact working. And heading into potentially my last field season this summer, I'll continue to replicate the survey locations and bring in new sites to ensure that these patterns are robust. And so undoubtedly, the most important step is going to be integrating indices of habitat structure. I'm talking about canopy height or greenness, for instance. And I'm planning to process publicly available satellite imagery from NASA via that finessed grant that Robert mentioned in my intro, in my later chapters to improve the predictions of this model and hopefully describe the causal relationships at play. So now transitioning to chapter 2, which is more or less a side project, really a natural byproduct of our field sampling these past 2 years. In recognizing the inherent complications of collecting physical blood samples, I wanted to attempt to improve the way we were collecting mercury data in the tropics by asking, are dried blood spots a reliable tool for monitoring mercury bioaccumulation? And so venous whole blood is sort of the gold standard for monitoring mercury bioaccumulation in organisms because it represents dietary mercury exposure within the past few days or few weeks. And so it's therefore the best way to estimate contamination at a given site. And that's really important. But blood sampling comes with its own set of challenges. We require a lot of bulky, expensive equipment like coolers and vacuumtainers to collect whole blood. Most annoyingly, we need to freeze whole blood prior to analysis to prevent coagulation in our samples. And to do this properly really requires a portable freezer and generator on the right, which really still struggle in climates as hot as Amazonia. And so we've gotten to the point in the global north where we sort of accepted this as a status quo, but in remote tropical locations, this becomes incredibly difficult and expensive to execute properly. And so dried blood spots may give us an opportunity to eliminate some of those issues in being able to store whole blood samples at ambient temperatures. Now, leveraging the existing technology of Whatman 903 protein saver cards, I've been developing a simple, user-friendly method to analyze blood mercury samples, specifically by accounting for the volume of blood that we're placing on the cards rather than the mass, which is typical in whole blood analysis. And so to validate this method, I've been collecting paired samples of whole blood and dried blood spots from a few hundred individuals while at the bird banding table. And the pipeline looks like this. In panel A, we use brachial venipuncture, as we typically would, to extract venous whole blood into a capillary tube. In panel B, we are measuring the distance that the blood takes up in the tube using a standard banding ruler. In panel C, we are drawing the blood spot onto our Whatman card. In panel D, we are cutting out that blood spot and feeding it into the DMA, or direct mercury analyzer. And lastly, and really panel F is the final step and the most crucial step because we are converting our blood mercury distance to a blood mercury volume using the volume to length ratio of the capillary tube model. And we'll then use this as the denominator of our mercury concentration. So I then built a simple linear mixed effects model in R to quantify the relationship here between the concentrations of our paired whole blood and dried blood spot samples, which were analyzed on 2 different DMAs. And I also included site and species as a random effects intercept to account for pseudo-replication. And so just as expected, we're detecting an extremely strong correlation in R squared of 0.97 between these paired data. And our predicted mean, but also our 95% confidence interval are both falling within the US EPA standards for environmental sample variance of plus or minus 20%. So that's a huge win. And I want to highlight Luis Wamani, who has been absolutely instrumental in generating these data while working in Cincia's lab. So gracias a Luis. This was hardly an analysis, but I did confirm that the equipment costs were essentially identical between this method and traditional whole blood without the costs of refrigeration. And so if you want to account for the cost of refrigeration, obviously your sample costs will go up, but right now we can sort of reduce the cost down to about $1.25 a sample, which falls under 5 soles in Peru. So we find dried blood spots for mercury analyses to be precise and reasonably accurate, replicable across DMAs, sites, and species. None of these factors had a significant effect in our model. Overall, these are cheaper than whole blood sampling equipment when considering the cost of refrigeration, but also the mental and physical burden associated with that. I never want to see any biologist in the future lugging out a heavy field cooler like this. They're also robust to tropical field conditions. I think it's important to state that I did not baby these samples. The dried blood spot samples that we collected were stored usually in my backpack at ambient tropical temperatures, basically until we got them to the lab space. And so with these results, I believe that we can utilize this method with a lot more confidence in future biomonitoring, especially in tropical regions, which should help scientists in the tropics operating on typically more shoestring-like budgets, limited budgets. So to distill some of the contributions to science here, we've developed an accessible tool that can support future monitoring. This should have a disproportionately positive impact for data collection in remote, understudied areas like many parts of Amazonia. And we will continue to generate this roster of biodiversity externalities for ASGM by leveraging the entire understory community rather than just individual species. And so especially given the current lack of effect concentrations for neotropical species. At the end of my PhD, I really want to be able to communicate a solid statistic that says with X% more mercury exposure, we can expect X effect on birds. And so regardless of whether that effect is positive or negative or even a null result, that's not important to me. But these are the kinds of hard figures that conservation biologists need to be able to move the needle on regulation of these industries. And so indeed, we are seeing a great push to transition from illegal and informal gold mining to more formal gold mining with the goal of creating more positive externalities or the reduction of these red arrows for human health, community infrastructure, and biodiversity under the United Nations Sustainable Development Goal Framework. So for centuries, humans have used birds as sort of these early warning systems for human and environmental health threats. And I hope I've convinced you today of the merits of focusing on mercury pollution as an underappreciated stressor of bird conservation in Amazonia. And I think that understory invertivores are incredibly well positioned to supply key data in the transition to more formalized gold mining, serving as our canary in the coal mine, or better yet, our antthrush in the gold mine. So I want to close this presentation by acknowledging the many people in Peru and also the US that continue to make this research possible, especially my field team. This is a photo of us last summer being escorted out of La Pampa by the Peruvian military. So thank you to their armed support in that case, as well as my funding organization. So my collaborators at Cincia are funded in large part by US aid. And due to the recent decisions of our current administration, they've been forced to pause all field operations until further notice. They've really been hit hard by this decision. And I'm currently fortunate enough to be in a position to fund a third and potentially final field season this summer. So I'd like to give a special thank you to AOS for continuing to support scientists during these times to ensure that good science can still move forward. And with that, I'll pause for any questions. Thanks so much.
Yeah, thank you so much, Chris. Fantastic talk. I feel like I can speak for the rest of the audience when I say that very impactful work. Both chapters have huge implications. I feel like moving forward. So I think we all really enjoyed that. Here's all the great jobs. Congratulations coming in the chat. I enjoyed the Mercury Olympics as well. I don't know if the first place would, I don't know if you would give a gold medal out though to the first place at the Mercury Olympics, but
It's amazing the things that conservation biologists do to keep ourselves sane, I
yeah,
think.
right. It's a little gloomy how it's going. The
For sure.
Olympics were a nice refresher. Yeah, many compliments coming in. I'm just going to just look for questions and just remember you can raise your hand and I can unmute you as well. Um, One person who is anonymous, it looks like, says, "Is there a reason the blood spots are drawn?" Okay, this is our question. Is the reason the blood spots are drawn as smiley faces in some of those pictures? So.
Yeah, so a side product from the one that I presented in chapter 2, I'm also simultaneously screening these birds for avian malaria infection. And so I'm usually ending up having to split that blood spot into two parts. So we take the majority of the volume and parse it out into a dried blood spot for mercury analysis, and the other portion is being screened, and we're going to sequence it in a molecular lab for malaria parasites. So I like to have fun with it. The design really does not impact the data quality at all. It's just a way to keep myself sane in the field, I think.
So I have a few questions that I will ask as long as I'm not missing any. Um, did you look at any predators? Like I didn't see that in the category. Like I think you were holding a forest falcon in one of the images, but was that a category in your graph?
It is, yeah. We actually only have, I think, two analyzed samples from a terrestrial vertivore. They fall solidly in that. So things like raptors, we only
Mhm.
have like two samples. So I didn't feel it was necessary to put it up there. But yeah, so basically along that trophic gradient, we have generally the sequence is we have granivores, then we have frugivores, then we have omnivores. Nectarivores like hummingbirds straddle the difference between an omnivore and vertivore and insectivore. And then higher than insectivore, but between insectivore and aquatic predator are those terrestrial vertivores. So they fall
Mm.
beneath the piscivores in this case.
Those two samples.
Yeah, but obviously not a lot of predicting power.
Mhm. And I'll just continue here, but anyone feel free to interrupt me. I noticed that some of the highest um, insectivore, low-lying insectivores, invertivores, were some obligate ant followers. Do you think there's any relationship between the obligate ant following behavior and mercury accumulation, or just happens to be that way?
For sure. Yeah. No, there's clearly a relationship there. You know, ant birds are typically performing, as a unit. Not all of them are obligate ant followers, obviously, but as a unit, Thamnophilidae, the ant bird family, is consistently performing in like the highest bracket in terms of mercury bioaccumulation. So there's clearly a relationship there. I don't exactly know what the mechanism is. I've sort of racked my brain for a long time about it. My sending hypothesis again is that these birds that are following the army ants, and as that army ant swarm moves across the forest floor, it's kicking up all of these insects, some of which are cannibalistic like spiders, and so these birds might have disproportionate access to those spiders via their following of these swarms. There's I'm not sure how much anecdotal evidence there is for this, definitely no quantitative data that I've seen, but as the army ants consume vertebrate prey items, things like lizards and things might have really high concentrations. If some of these ant birds are consuming those as bycatch, that could be spiking their mercury levels. So we definitely need to invest a lot more time and effort into being able to disentangle that mechanism. But yeah, it's really interesting and obviously concerning from a conservation perspective.
Thanks. And now a bunch of questions have rolled in. So I'll ask Sarah Lipshitz's question first. Did you observe any behavioral impacts in high mercury species individuals? Yeah, great question.
Yeah, fantastic question. It's not something that we have the capacity here in terms of our sampling design to be able to do, but I'm open to chatting about potential collaborations is what I'll say. In lab settings, and I also think in some amount of field settings, there have been significant relationships between mercury bioaccumulation in both lab reared and also wild birds, and mercury. And so there's probably something going on, but we don't have the capacity to observe it directly because we're capturing these birds passively and not really keeping them in hand for longer than 15 minutes or so.
And then Robert Pagliaroni asks, is there a tipping point or have we reached it already in the amount of mercury these birds can contain?
Yeah, fantastic question, Robert. We have to imagine that there is some sort of like biophysical ceiling for these birds, that feathers are used to depurate a bird's mercury body burden. If feathers are one of the primary pathways that a mercury can reduce its toxic load, and so part of the reason that feathers are useful is that it's essentially indicative of the amount of mercury that a bird was more or less successful in able to remove from its body. And so there definitely is a biophysical ceiling in the amount of mercury that they're able to actually press into the feathers. It, I would say most proximately would be the molt window, right? And so mercury is only able to bioaccumulate in feathers in the time that a feather is exposed to the bloodstream as it's molting out. And so once that feather stops growing, once the molt cycle is completed, that bloodstream to feather connection senesces. And so it's almost like a time capsule locking in that mercury. But yeah, I think that there's definitely a biophysical ceiling at some point. Whether or not we're able to actually detect it at the end of the study is a different story. It's sort of a guessing game at this point, but yeah, there's been a lot of lab studies that have shown that, you know, to produce an effect concentration of this amount of mercury yields mortality. And so that's I guess the answer to that question. At some point, we know that would be happening, but the question is whether those concentrations in the environment are high enough to be able to cause that. That's a different story.
A key question. And James Malta asks, I'm imagining that cost is the primary constraint in looking for a method for non-refrigerated sample analysis, and therefore an anticoagulant wouldn't make sense to use. Would anticoagulation affect the sample in a meaningful way, so long as the volume was taken into consideration?
So if I understand your question, Jimmy, these are paired samples, right? So we have it's a one-to-one comparison using the same blood. And so that one-to-one comparison is yielding a really high concentration via our paired samples. And so for what it's worth, I don't see an issue of the anti or coagulation effect at play in our samples. There is a slight bias towards dried blood spots actually being higher. There's a slight bias. It's not significant, but there's a slight bias that there's slightly high more contamination of dry blood spots than whole blood. I mean, the reason that I think that could be is either external contamination or internal contamination of the cards. There's really no evidence for that via our blank samples. And so what I'm thinking is potentially at play is that, you know, at some point in your whole blood samples, clots do happen. And so sometimes those red blood cells stick to the interior of the capillary tube wall and are not expressed into the sample that you feed into the direct mercury analyzer. And so that could be causing almost a negative bias in the amount of mercury that we're detecting in those samples because we know that methyl mercury binds to red blood cells. And so yeah, hopefully I answered your question there.
I'm not 100% sure we're going to get to every question. There's a bunch of questions still. Jennifer Amaya says, great work, Chris and field team. You mentioned that you deployed mercury passive samplers along these transects. Did you find any trend with the mercury content in birds with those?
That's a great question and something that I haven't had the opportunity to look at yet. We expect that there could be, whether or not there's enough variation, I guess, along the passive air samplers to correlate well with the bird concentrations. We expect that there could be. Generally, I know generally the results of the passive air samplers. I just have not received those data yet from the lab. We are detecting seemingly higher mercury in the airspace in adjacent to or directly in artisanal mines as compared to control sites. But yeah, I haven't looked at that correlation yet, but hope to in the future.
And Chris Anna sent a message that we can go a few minutes over to address these questions. So if everyone sticks with us, uh, uh, we don't have to rush through any answers. Fami Ferdo, if I said that correctly, says, "Hi Chris, does natural disaster affect the mercury accumulation in the bodies of birds? For example, floods that bring more minerals and pollutants from the mining sites to the forests."
Absolutely. We imagine that it does, both in flooding regimes, you know, that can bring nutrients, which can stimulate methyl mercury production, but also it can bring inorganic mercury onto the terrestrial landscape where it can then be methylated by soil and detrivore communities. Um, Another natural disaster to keep in mind is actually forest fires, which are in general increasing throughout the Amazon basin as um, land clearing increases. And so trees are great sequesters of carbon, but also mercury. And so as trees burn, it's emitting that inorganic mercury back into the atmosphere. Those re-emissions are actually pretty insane in magnitude. So yeah, that could be recycling that mercury in the airspace continuously every dry season, essentially.
Okay, and Natalia Ocampo asks, what are some mitigation options that exist to counter these mercury effects? Yeah, some solution. Mhm.
Yeah, definitely a poignant question. I so the primary mitigation option, I think, for mercury reduction, generally we have usually they are engineering sort of fixes, right? And generally there are solutions that exist. One, by and large, is a retort. It's this like in the cheapest sense, it's a metal pipe that has one end you can open, the other end remains closed. You like heat that. You put the mercury gold amalgam inside. You then burn that open. It vaporizes the mercury, but leaves behind the gold. But it's really the social cultural uptake of those engineering fixes that has lagged behind, right? So there's sort of this, at least in the literature that I've read, and the conversations that I've had thus far, the mining communities have been slow to uptake those engineering advancements because there's sort of this like cultural bias or understanding that it somehow tarnishes the gold. There's like it's a less uh, the quality of the product is less, and so they don't want to use it. And so that's really the major hurdle I think moving forward is to offer up cheap, scalable engineering fixes to reduce the amount of mercury that will still yield a similar gold product that the miners want to produce.
Very interesting. Uh, just because people are we're wrapping up, I'm just going to put the post seminar evaluation form in here one more time as a reminder for everyone. And lots of great questions. Thanks so much. I think I got through all the questions here in the various ways that people can ask. Um, and we just want to thank Chris so much for the great talk, for answering all of our many questions, little grilling session, great answers.
Hardly.
And no, and we really, at least I really appreciate being educated on something that I did not know much about previously. So we want to thank you so much, Chris. And we will see you guys all here next week for the next seminar. So thank you all so much for being here.
Thanks
Bye,
all, and thanks
guys.
for